All right. Good morning, guys. I hope everyone is fine. So let's start. Remember what we said last time is the structure of the eye. We saw the structure of the eye, and we did some activities about the eye. Yes, and we say that one of these activity we did, we say that if you look at A, we said, can I have answers now? Let me just have answers on the labeling. After that, we can go to the mechanism, that is Pupillary mechanism and then accommodation. What is A?
Ciliary muscles.
Ciliary muscles. B?
B is suspensory ligaments.
Let me clear it up so that you're able to see. Yeah, what is B
B
now?
the iris.
Is the
B is
iris.
suspensory ligaments. It's not pointing at the iris.
The suspensory ligaments are this. See?
Oh,
See, let me
okay.
zoom it out so that you're able to see exactly. You see that B is focusing on this. Are you seeing?
Oh,
So
yes,
that
sir.
part is the iris. So the suspensory ligaments are these threads you see. You see these threads? Yes. So those are the suspensory ligaments. So B is the iris. Thank you. Then what is C?
Lens.
Lens.
The lens. F.
Lera.
F is
Or
the
sclerotic
sclera.
muscles. What is E?
Blind spot.
Choroid. Choroid.
Choroid.
Choroid.
Yes. And then what is D?
Optic nerve.
Optic nerve.
F. And then what is the O?
Blind spot. If
Blind
not the
spot.
yellow. If not the yellow
Blind
spot.
spot.
then it's the-
It's a yellow spot. That's where the clearest image is formed. What is P?
P is the retina.
Retina. Talking about that part which bends like that.
Yes, sir. The retina.
It's the retina. That part which bends there. We call it the blind spot. That's where the nerves are coming from. Let me show you what I'm saying. Talking about this part. You see this depression? That's where the nerves are coming from. It's what you call the blind spot. So the retina is this white part you see. This part. See? This, yeah, that's what you call the retina. This part which you see I'm trying to shade is the retina. But where the nerves are connecting to the retina, yes, that part here is what called a blind spot. So that is a blind spot. Then, then how do you call this part of the eye? How do you call this part? This part which I'm covering. How do you call that part?
The vitreous humor.
Vitreous humor. And then how do you call the front chamber of the eye? This part.
Aqueous humor.
Yes, humor. So now you need to know each and every function of the parts which you have tried to mention. There is a space here between the two iris. How do you call that space?
The pupil.
The pupil. Yes. So that is the simple structure of the eye, and we saw it properly when we are talking about the labeling. So now we need to know what we call pupillary mechanism. Pupillary mechanism. Okay, here starting with accommodation, but it's fine. Let's look at the pupillary mechanism. When you look at the pupillary mechanism, don't forget that this is also a revision. but let's just do a recall of it. So remember that the eye has what you call the lens. The lens is held in place by what you call the suspensor ligaments. On top here, you have what you call the ciliary muscles. But an extension of it forms what you call the what? The iris. So if you look at this iris, the function of the iris is to adjust the space. Either the space to be bigger or the space to be smaller.
Smaller.
The ability of this iris to change this space, which we call the pupil, is what we call pupillary mechanism. So it comes from the word pupil. So it becomes pupillary mechanism, meaning that this is the eye's ability to be able to see basically in dim and bright light. But how does it see in dim and bright light? It's by the iris allowing the specific amount of light into the eye. How does the iris do that? By adjusting the size of this pupil. So when the iris adjusts the size of this pupil, so we call it pupillary mechanism. The question comes in, how does the iris able to adjust this space A and the space B? So the iris has muscles. It has the muscles which run in the circular way. They run in the circular way. So we call those muscles the circular muscles. The circular muscles. So it runs around like this. Né? So those muscles are called circular muscles. Then it has other muscles which run like this. They run like this. I mean, if you look at it cross-sectionally, you'll see that these muscles, they run like this. So these muscles, because they run from the circumference going towards the center of the iris, this distance is called a radius. So it means that we're going to call them radial muscles. So the muscles which run like this, we call them radial muscles. While the one which run in the circular way, we call them circular muscles. So now, how does the eye bring the adjustment, and how does the iris brings about the adjustment of the space using these two muscles? First of all, imagine the iris is a banana. Yeah, imagine the iris is a what? Is a banana. And then now you're holding the banana like that. So if you're holding the banana, you try to squeeze it. What muscle are we going to be squeezing? Which muscles are going to be contracting? You'll find out that the muscles, if you hold a banana in your hand, you see that these are the muscles which you are going to be squeezing. Meaning that it is going to cause this banana to extend. If this one extends and also this one extends, it means that now the distance between the two bananas, you are pointing them each other, is going to reduce. Meaning that that's how the iris also works. When these muscles, these ones, it try to contract, they push the iris to each other, and then now the space between the two closes. When the radial muscles try to pull this, meaning that they are pulling this side and also pulling that side, meaning they are trying to contract, it means that now they're going to make the space between the two, this and this, to be bigger, meaning that now the iris, the pupil is going to dilate. So how do I remember this in a simple way? I always say, you say this, that I'm bright like a star, I don't mix things up. During bright light, things are not supposed to be mixed up. For example, if you have light and we give you food to eat, you will know where to put the hand and where to put it from the plate to the mouth. Yes, the mouth can be fine, but the plate, you won't miss what you want to pick on the plate. But imagine when we remove the light, meaning that you won't be able to see exactly where you're putting the hand. So we're going to call it, meaning that you're going to be mixing the food. So since we have letters, which is R, which we say that R is capital, R is the radial muscles, and then you have the letters C, which will mean sacral muscles. We have another letter R, which we call relax, and then we have another letter C, which we call contract. So if I don't mix things up, when don't I mix things up? That is during the bright light, meaning that in bright light, I don't mix these letters. So the R will go with the R, that is bright light, because I don't mix them. R will go with R, and then the C will go with the C. And I will say that this is bright. This is bright light. So meaning that in bright light, R will go with the R, C will go with the C. But remember, in bright light, I will require less light entering because it's too much. So it means that my pupil will be smaller, meaning that if you look at these muscles, yes, my pupil will be smaller. What about in dim light? Dim light. Dim light, I mix things up. R will go with the C, while C will go with the R. You understand? Meaning that because it's dim, I will open my eyes so that I'm able to see clearly. So it means that my pupil is gonna dilate. See, my pupil is gonna dilate. So it means that in dim light, the pupil dilates. It dilates. While in bright light, the pupil will constrict. So since I know that, I will go back and write. During bright light, radial muscles relax while sacral muscles contract. The diameter of the pupil will constrict, meaning that less light will enter the eye. While in dim light, the radial muscles will relax. Sorry, the radial muscles will contract while the sacral muscles will relax. Well, how about the pupil? The diameter of the pupil or the pupil will dilate, meaning that the pupil will become bigger. And then now more light will enter the what? Will enter the eye, because this is dim light. So I will advise that you memorize one. If they bring bright light, if you have memorized the dim light, then it means that you're going to have to talk about the difference or the opposite. If this bright light, radial muscles will relax, so in dim light, radial muscles will contract. If in bright light, sacral muscles will contract, in dim light, the sacral muscles will relax. So it means that you don't need to waste a lot of energy. So you need to know, maybe if you are going to be confused, make sure that at least you memorize one. So when we go back to our booklet, we see that radial muscles relax when in the bright light, while sacral muscles will contract when in bright light. Check. The pupil will constrict. It means that it becomes smaller, meaning that the diameter of the pupil will constrict. It will become smaller. So in science, we're going to call it constrict. Yes. If it constricts, what do we say? We say that less light is going to enter the eye. So in this case, less light is going to enter the eye. So that is pupillary mechanism. Radial muscles, yes, relax. Circular muscles, contract. Yes, the diameter of the pupil will be small, meaning that the pupil will constrict in actual sense, and then now less light is going to enter the eye. What about, that's why we say that R with R, C with the C. In bright light, don't mix the letters. R will go with R, C will go with the C. That's it. And then if you look at them here, you can also see it that the diameter of the pupil has constricted. If you compare this pupil size and you compare this pupil size, if you compare this one and you compare this one, you'll see that this is small, while this one is big. Yes. So in dim light, we said we mix them up. R will go with the C, while C will go with the R. So radial muscles contract, circular muscles relax. The diameter of the pupil will dilate, meaning that pupil is going to become bigger. And then now more light will enter. More light will enter the what? Will enter the eye. So that is radial muscles contract, circular muscles relax. R with the C, C with the R. So you see that now they can bring this. They can bring these diagrams, they can bring graphs. I saw here a question. Let me show you. Let me show you a question here. It was a graphical work. Check. Change in the pupil size. That's the diameter. You see, if this is the diameter, then it means that at here it's 6, while at here it is 2, meaning that here the diameter of the pupil is large, while here the diameter of the pupil is small. So it means that if you look at here, maybe this person was viewing in dim light because the diameter of the pupil is what? Is at 6, is bigger than the diameter of pupil when it comes to 30 seconds here. It means the diameter of the pupil is around 2. So meaning that this was bright light. What about this? Maybe it was a moderate light because sometimes the light is not that too much, it's not that too small, so it's just moderate. Then it means that even the diameter will adjust according to the amount of light which has been subjected to the eye. Yes. So that is pupillary mechanism. Let me just get one person here to tell me if it's clear. Chico.
Hello.
Is it clear?
Yes.
All right. So you guys, if you have a challenge, you can raise up the hand. I hope you know how to raise up the hand using this. If you can't know, if you don't know how to raise up the hand, you can unmute and you say, excuse me, so that I'm able to hear that you have a concern when we are going to, or when we explain a specific concept. All right. So that is the dim light, bright light. That is what we discussed last time. Let's now discuss about the accommodation, and then we see, just to do a review of most especially those people who were absent, and then yeah. All right, let's go to accommodation. Before we go to accommodation, you need to note this. When you are talking about dim light, bright light, you only need to talk about the iris, the pupil. That's it. Don't talk about the lens. No. Accommodation. In accommodation, we say that our eye looks like that. You have remember we said that remember we said that, and then you have the suspense, sorry, this ciliary muscles there. So if that is my diagram, we say that this is out now because this was talking about pupillary mechanism. So now what you're talking about now is accommodation. We need to talk about the ciliary muscles, suspensory ligament, and then the lens. Then we also talk about the retina. So those are the 4 things which you need to talk about when you talk about accommodation. So what is accommodation? Basically, it's the ability of the eye to view the near and the far object. How does that eye do that? It does that by adjusting the focal length. Yes. So how does it adjust the focal length? These muscles, they must relax and contract so that it brings about that. So, how does the eye see? It will classify the vision into 2. That is near object and far object. Any object which is 6 mirrors and above, we call it a far object. Any object which is less than 6 mirrors, yes, less than 6 mirrors, meaning that, they write it like that, less than 6 mirrors, then it means that that is a near object. But what happens when you are viewing a near object? Let's go back to the simple concept. I am sitting here, a plane passes on top. Yes, a plane passes on top. Just a moment, guys. Just a moment, something. This thing, I don't know what's happening here. Just a moment. Yep, I'm gonna come back. I'm gonna come back. Okay, let's do like this again. There are some colors which have just happened on the screen, so okay. Yeah, back there. And then okay. All right. Okay. We are around there. And then it. Right. Yeah. Okay. Zintle, are you in class? Okay. Let's continue now. You're saying that to talk about accommodation, you need to know this. You need to know this. Number 1. Is that?
So your mic is having interference.
Sorry?
Your mic is having interference.
Yeah, I'm looking at that person who's what's happening. Let me just, guys, make sure that you mute. Yep, think it was that person. Yeah, mute. Good. Yeah, it was someone's mic. So now, okay, we come back. Now, to talk about accommodation, we say that abilities eye to see, to focus on the near object and the far object. And we say that a far object is 6 mirrors and above, and the near object is less than 6 mirrors. So now, we say that, take an example of an aeroplane. You are sitting somewhere, and then an aeroplane passes there in the sky. How big is an aeroplane? It's very small. Meaning that if these are the parts, let me just do like this. These are the parts which are, you have two kinds of lenses. When the lens is flattened like that, we say that it's less convex. If the lens is thick like that, we say it is more convex. Understand? So if this is our retina and this is our retina, you have the suspensory ligaments holding the lens to make this lens thin like that. This suspensory ligament must pull the lens in opposite way, so that it's like you holding a balloon and then you pull the two sides of the balloon. It becomes flattened. That's what exactly what happened to this lens. It's being pulled, so that's why you see that these suspensory ligaments are straight. It's not a mistake that I drew them straight. In science, we call this tau. It has become tau, meaning that it has been stretched. It is stretching it. Then here we shall say that the suspensory ligament will have that S shape. In science, we call it slacken. So these suspensory ligaments, they have a muscle which is attached to them. They have a muscle which is attached to them. That's what we said. This muscle, we call it ciliary muscles. Ciliary muscles under suspensory ligament, they work in opposite way. In science, we call it antagonistic. They work antagonistically in opposite way. Meaning that if this one contract, this one, the suspensory ligament, must relax. If this one relax, the suspensory ligament must contract. But don't forget, we don't use the word contraction on the suspensory ligament. So we shall say that if the ciliary muscles contract, the suspensory ligament must slacken. Meaning that if you see the suspensory ligament slackened, then it means that the ciliary muscles are contracted. If you see that the suspensory ligament are straight, it means that the ciliary muscles are relaxed. Meaning that the suspensory ligament have been slackened. I hope you guys, you're understanding the concept by concept. So you go back and also watch the other previous video, and then you combine the two so that the eye is no longer a problem. Okay, now we continue the next step. We say that when you look at the aeroplane in the air, you see it as small. When you look at the aeroplane at the airport, those who have ever been at the airport, you see it as huge. Meaning that if an object is near, it is going to be seen as huge. Meaning that I will require a big lens, a thicker lens, to refract, to bend this light, which is coming from this big object, to fit it into the eye, to the retina. If this is the aeroplane, if that is the aeroplane, this aeroplane is near, all this beam is supposed to be converged at the retina so that you are able to view it. Meaning that if I see an object which is near, I will require a bigger lens, a thicker lens, to focus it on the retina so that all this big object you are viewing, it must fit into the retina. Or about when this aeroplane is too small, is far, you see it as very small. Check the kind of beam which is going to enter the eye. Meaning that because the beam is too small, then I will require a smaller lens to bring the image onto the retina. So when you look at the object and you see as big, then it means that the lens must become more convex. If you look at the object and you see it as small, then it means that the lens must become less convex. So it's a simple procedure. Near object, the object is big. Therefore, the lens is supposed to be thick. So now the question comes in. How do I make my lens thick? It means that the suspensory ligaments must relax, but we did not use the word relax. We say that it becomes slacken. Then the ciliary muscles must what? Must contract because they work in opposite way. So now I have a story to say. In nearby object, okay, step 1, I will start with this. We say that the ciliary muscles will contract, the suspensory ligament will slacken, the tension on the lens will decrease. The tension on the lens will decrease. Okay, if the tension on the lens decreases, it means that the lens will become more convex. The lens will become more convex, yes. And then now, meaning that now the refracting power, the ability to bend this light is more. The ability to bend this light is going to become what? It's going to become more. Meaning that now the light will bend more to the retina, and then now a clear image is going to be focused onto the retina. So that's the meaning. So it is said that step 1, the ciliary muscles will contract, the suspensory ligament will slacken, the tension on the lens will decrease, then now the lens will become more convex, meaning that it is going to become refracting power is going to increase. The ability to bend this light is going to increase. So the lens becomes more convex, and then light rays are bent more onto the retina, and then now a clear image is focused onto the retina. So that's what we are talking about here in our booklet, that a nearby object, check, these muscles are not mistakenly made like this. No. Because if you look at that and then you look at this, you see that these ones are straight. So meaning that this one now, the suspensory ligament are taut. While here, the suspensory ligament is slackened. So it means that it's not by a mistake that this is happening. It's not. It's not by the mistake that it is happening like that. So ciliary muscles contract, suspensory ligament slacken, the tension on the lens will decrease. When the tension on the lens decreases, it means that now the lens will become more convex. It becomes more thicker. Now the light rays are reflected more, meaning that they are bent more. And then now a clear image is focused onto the retina. That's what we call accommodation. And that process will result in the change of the lens so that a near object and a far object can be focused. Meaning that if you don't have ability to change this lens, you can either be a short sight or you're going to be a long sight. We will see that. So now this is the opposite of a nearby object. We go back here. A far object, the beam is small. So meaning that the lens is supposed to be less convex. How do I make it? By pulling these suspensory ligaments. When I pull the suspensory ligament, in science, we call it become taut. And then what about this? It's gonna relax. So the ciliary muscles will relax. The suspensory ligament will become taut. The tension on the lens will increase, meaning that the lens becomes less convex, meaning that light rays are bent less. And then now a clear image is focused onto the retina. Why? Because the object is small, is far. We see it as far. We see it as small. In actual sense, it's not small, but it appears small. So that's what we are saying here, that the ciliary muscles, please. This is the major mistake students do. When you ask them accommodation on the pupillary mechanism, when they reach accommodation, they will write, they will say, sacral muscles will relax. Sacral muscles in accommodation. You always make that mistake. Sacral means circle. It's only the iris which has the sacral muscles, the muscles which are moving around. So you have to know that. Don't mess it up. I repeat, don't mess it up. Yes. So guys, ciliary muscles will relax. The suspensory ligament will become tightened. That is taut. Yes, the tension in the lens will increase because you are pulling them in opposite direction, and then now the lens will become less convex. And then once it becomes less convex, then the reflecting power will decrease. That's why you're saying that the light rays are refracted less, and then a clear image will be focused onto the retina. So you're able to see a clear image here onto the retina. So you guys, I've spent a lot of time there, and then I think we can go to the defects of the eye. And I ask someone here. Yes, Ariel. Ariel, I'm not going to ask you, came late. Roy, Royce.
Yes, sir. Yes, sir.
Are we together?
Yes, sir.
How do I pronounce your name?
Voice.
Royce. No, okay, thank you.
Yes.
All right. Pirason, Pirasen, are we together?
Yes, sir, we're together.
All right, all right. Okay, since we are together, now let's shoot to the next part, which is the eye defects. Eye defects. We have 4 kinds of eye defects. The first one, before I talk about eye defects, guys, please don't go to the paper without knowing pupillary mechanism and accommodation. Don't make a mistake, because that mistake will result in loss of marks. Eye defects. Eye defects, we have the short sight and the long sight, astigmatism and cataracts. It is simple. When you say short sight, what does it mean?
We don't see something.
Sight, meaning that you are able to see objects which are in short distance. You are short sight. When you say long sight, it means that you are able to see objects which are in a long distance. So short sight are those people who are able to see objects which are near, but they can't see objects which are far. That is the most common eye defect. We see objects which are near, but we can't see objects which are far. So we call it short-sightedness. That's what we call it. Then, in science, we can also call it myopia. Myopia. And then long-sightedness is you are able to see objects which are far, but you can't see objects which are near. So we call it long-sightedness. We call it long-sightedness. Then we have another sight which we call astigmatism. Here is very common in older people. We're going to see what causes that. And then the last one, we have another one called cataracts. This one is common in any age, but we will see what causes it. Let's go to the causes of each. Short sight is caused as a result of the eyeball is too long. If the eyeball is too long, what does it mean when they say that the eyeball is too long? You're able to see objects which are near, but you can't see objects which are far. Yes. Remember, you have, okay, let me use this diagram. Remember, the eye, objects which are near, the image is supposed to be focused onto the retina, meaning that even this eyeball is big, this thing is big. So now the lens is thick. You understand? Is thick. Listen, and I show you that how does the eyeball being too long, what does it mean? Where does it come from? But now, because you are seeing an object which is near, we say that if you are seeing an object which is near, the lens is thick enough. You can't see now the object which is far. Guess what? This lens cannot just a smaller size. It cannot do this. It cannot change from thick to small. Now, you want to bring this light which is coming from from far, and you want to use a lens which is thick. It's like you want to kill a mosquito and you bring a hammer. Meaning that the mosquito will die and even disappear. That's the power of the hammer over the mosquito. So you are bringing a thick lens to be applied on a small beam, meaning that this light, when it comes like this, too small, it's gonna be bent very much before it reaches the retina. Meaning that the eyeball is too long for this. That's the meaning. This light couldn't reach the retina. It's too long. It's like they ask you to jump the river. Just jump by jumping. However much you know how to jump, you cannot jump. You end up falling into the what? The river. Meaning that the river is too long for your jump. It's fine when you are using a plane, you can jump it because the plane is strong enough to jump over the what? The river. Do you understand? So this lens is designed for big beams. But now you bring this small beam. Obviously, it will try to jump, but it will not manage to cross over. So it means that it will be just in the middle there. That's why they're saying that the eyeball is too long. That's why now you see the image is focused before it reaches the retina. The image falls before it reaches the retina. Number two, the cornea is being too curved. Meaning that if it is too curved like this, it means that now it is more convex. The light is being bent too much. It's being bent too much before it reaches even the lens itself. So it makes basically the image to be focused before the retina. So what do? We have two types of lenses. We have a lens which is a concave lens. We call it a diverging lens. And then you have another one which we call a convex lens, which we call a concave lens. Because now we want to push this image to move from here, this point, to the retina. What am I going to do? I must make this light which is coming act like is coming from a nearby object. So I have to put a diverging lens so that when this light comes, it's going to be diverged a bit, become a bigger beam. So by the time it falls on the lens, now it is bigger. The beam is bigger. And then now the image can be focused onto the retina. The same story here. If I want this light to reach here, I have to apply here a diverging lens so that now this light is going to come. By the time it reaches the lens, it is coming in this direction. You see, it has been diverged. You see, it has been diverged. When it reaches the lens, it will appear as if it is coming from, a nearby object. Now this lens can use its power to diverge it onto the retina. That's why you're able to see. Meaning that people who are short sight, they have to apply a diverging lens, which we call a concave lens, so that they're able to see the things they see, which is far. The eye will see it as a mere object, because that those glasses they put on, they have a diverging what? Lens. So that is short sight, and is the opposite for long sight, meaning that the eyeball is too short. And then now, this person is able to see objects which are far, but you're not able to see objects which are near. The eyeball is too short. Remember the near object? What happens? The far object, the lens is too small. So if you are able to see objects which are far, but you can't see objects which are near, it means that the lens cannot change to be thick when the image is coming from nearby object. Check. The lens is too weak to focus the image onto the retina. So it means that the image is focused behind the retina, meaning that the eyeball is too short. So if the eyeball was there, then it would have been fine because it's long enough, but it's too short. So the image is focused behind the retina. So what is the next step? So it means that I will have to put on a convex lens. Convex lens, it's gonna make this light be converged. By the time it reaches the lens, it will be as if it is coming from a far object. And then now this light can be converged onto the retina. So that's why here we shall say that to solve that, the eyeball is too short or the cornea is less curved, meaning that it cannot convert this light. So how do we solve that? So we solve that by putting on the convex lens so that now the light can be converged. The light can be converged. That's why we're saying that you wear glasses with convex lenses. So guys, you need also to read about that. These questions usually come in objectives. And then lastly, but not the least, is astigmatism. Astigmatism is as a result of you form multiple images. The person sees the individual, but they cannot see an individual clearly. Why? Because the cornea is unevenly distributed. Look at the cornea. It's supposed to go through straight, but now it has these curves. So that some light is going to be going that side, some light is going to be going that side because it's not smooth. It's like you're driving on an uneven road. How is the car going to be? Meaning that this one goes this side, this one is pushing it this side, this. That's why it shows. But when it goes to the tarmac, which is smooth, it will start meaning that it will be moving in one direction. You see, nice. So it's the same thing. If the cornea is unevenly distributed, now the lights will be going in different directions. And guess what? You're going to form multiple images. How is that possible? Get an example of a mirror. Very smooth, one mirror. Look into it. Can you please mute? So we are saying that one mirror. When you look into your when you look into the mirror, you will see one image. You see yourself as one. Make a mistake and break it and you leave it intact. Break it and you leave it the way it is. Then check yourself again. How many people you're gonna see? You're gonna see multiple images there. It's exactly what this cornea is doing. If it is unevenly distributed, it's like a broken mirror. That mirror will gonna form multiple images because now light comes and then it's in different directions and then you form multiple images. If you look, you fail to focus. Which one are you gonna focus on in the broken mirror? Which image are you gonna focus on? Maybe you're gonna look at one of which is big enough and what if all of them are the same size? So it means that this is very common to people who are old. And this, in most cases, causes headache. Yes, it causes headache. Your go when you go Mukulu, you say, oh, it's you, Parabo. Oh, yeah. Then touches you to remember all the feelings and okay, yeah. But he says that the person, there is an object which is in front of me. So those people, they see you as an object, a big object which can talk, but might not be able to recognize your face. But because you speak, knows you very well, then he's able to know, oh, you are Rabo. Oh, you are Peterson. You understand? Yes. Huh. So we go to the next point, which is the cataracts. So how do we treat this? We have to put on glasses with the glasses which we call the spherical lenses. They have spherical lenses. They have equal distribution. Or you can go and then they do the laser therapy. What they do is if your thing is like this, so if your cornea is like that, so what they do is they cut off this part so that now the cornea is smooth. Just the smooth out and then the smooth in, so that now you're able to see properly. Cataracts. This one is as a result of the lens becomes cloudy. It becomes, it solidifies. It becomes opaque. The light cannot go through. It cannot penetrate. And if you look at it here, yes, the lens, this light cannot penetrate. It cannot go through. This lens is now opaque. It cannot allow, it's not transparent anymore. You understand? So it means that the light just hits on the lens and then bounces back, meaning that the image cannot be focused onto the retina. So people who are suffering from cataracts, you just have to go for a surgery and then they remove this lens, then they replace it with a synthetic lens. Yeah. So basically that is it. Let's do this question together. Yes, guys, let's do this question together. Let me do like this. I'm gonna give you 2 minutes for you to do this question. And then after that, we discuss about it. Then after that, we go to the ear. Don't forget to label each and everything. You label A, B, C, D, E, everything. You must label it. So I'm giving you 2 minutes so that you're able to answer that. Hey guys, let's try to answer the questions. Okay. They're saying, what is A?
The iris.
The iris. What is B?
The choroid.
The choroid. What is... just a moment. What is C?
Sclera.
Sclera. What is D?
Retina.
Retina. What is E?
Optic
Nerve.
nerve.
F.
What is E?
The optic
Optic
nerve.
Optic nerve. Guys, don't forget that this is a sensation. We discussed on the brain that all sensations go to cerebrum, meaning that this eye will send the impulse to cerebrum. Explain the effect of the person's vision if it's cut. It's now. So what will happen to the person? Will the person be able to see?
No.
No.
So explain the effect. Who can explain to me? Yes, Rose.
The impulse from the retina will not be able to be sent to the cerebrum, preventing the person to be able to see.
The impulse will not be sent to
The
cerebrum,
cerebrum,
preventing the person from
to be able to see.
See. Yes, ayandiswa. Ayandiswa. Yes, what I say?
So I said the visual stimulant will not be transmitted towards the cerebrum visual stimulant will not be transmitted, and the person would be therefore blind as the cerebrum cannot comprehend the sensation.
Yeah, so basically here, guys, thank you. Basically, we want to see that the impulse will not be sent. Where? Which part of the brain? We need to see that part of the brain. You understand? Don't just say it will not be sent to the brain. We'll ask you which part of the brain. And then and what will be the effect on the person's vision, meaning that the person will not be able to see. The person cannot see, meaning that the person will be blind and has no vision. Why? Because you see now, what are they saying? Because the impulse from the retina cannot be transmitted to, you see, if you don't just, if you don't talk about you have to talk about which part of the brain. Don't just say brain. Those people, if you have just written brain, yes, you have to mention the part of the brain where the impulse is going. Then they're saying that describe how part A, okay, how part A helps to protect part D. Part A protects. Yes, love. Yes. Yes, I am this one.
In bright light, radial muscles of the iris relax, circular muscles of the iris contract, the pupil constricts or becomes smaller, then therefore less light enters the eye.
Yeah. So basically here, they're asking you part A is the iris. It will help to protect the part D, that is the retina. Remember, retina is sensitive. So in bright light. So basically, what will happen? They're asking you the events which will happen in the bright light. Yeah, so thank you so much. Yeah, your answer is right. The circular muscles of the of part A, that is the iris. Yeah, we will do what? Will relax. Uh, oh, yeah. I'm here because I was saying relax. I was just reading here. It's not relax. The circular muscles, yes, of the iris will contract, while the radial muscles will relax. Yeah, remember it's bright light. C goes with the C, and the R goes with R. Yes, and then now the diameter of the pupil will become smaller, meaning that it will constrict, and then now less light will enter the eye. That's where the protection will be happening. So explain in the condition called pre-myopia, lens lose its elasticity. If the lens loses its elasticity, therefore it maintains a constant flat shape. Yes, if you are flat, meaning that you are not more convex, meaning that you are just flat like this. You see, you can't become more convex, thicker. So explain how this condition would affect the person's vision. Yeah, who can tell me the answer? Zinhle, you no longer giving me answers. Why? Are you there?
I'm not in the right space of talking.
Oh,
Yeah.
okay. Michelle, do you have anything to say? Michelle, do you have anything to say? Oh, the mic is a problem. Okay. Yes, we are saying that in accommodation, this is basically accommodation. Remember that you are able to see changing the shape of the lens is called what? Accommodation. And then now, if there is not, the lens is now, it cannot become elastic, meaning that it cannot adjust. Therefore, it means that accommodation will not what? Take place. You understand? And then if the lens is flat, it means that it cannot become thicker. And remember, to be thicker, you're able to see objects which are near. And then to be thin, your objects which are far. So when you have looked into a far object, meaning that there will be a problem in an individual seeing near objects because the lens is only small. It cannot be more convex. It cannot be more thicker. That's why you're saying that the reflecting power of the lens is going to be low, meaning that this lens, remember when you are talking about this, so it's going to be like this. It won't be able to be like this. So it only be like this instead of if it's viewing a nearby object, so it means that it will be like this. So now they're saying that a reflecting power is going to be low, and then what happens? Light rays are not refracted more or bent enough. They are not refracted more, and then that it means that it will not be able to focus onto the retina, meaning that that is for objects which are near. You understand? Therefore, image which are near, they will be the image will be focused behind the retina. Yes. So, meaning that if this is the retina, the image will be this side instead of being here. So now they're saying that therefore the person cannot focus objects that are closer. You see, you cannot focus objects which are near. You can only see objects which are at a distance. Why? Because the lens is less convex. So suggest the shape of the lens that may be prescribed by the doctor. So it means that then if your lens is only like this, meaning that I have to put on a converging, not a diverging, because you can't be able to see objects which are near, meaning that you're going to have to put on convex lens so that now it brings the light closer to appear as if it is coming from a nearby object. That's why they're saying here it is a convex lens, or what you call biconvex, meaning that if it is convex, just convex, it can be like this. Biconvex, it means that it is double-sided. You have this side and you also have this side. So that's the difference between convex and biconvex. It means that it's curved 2 times. Okay, guys. Let's talk about the ear. Ear is simple. It's only have as one issue. Just maybe the mechanism. Ear. Have you ever thought of why your ears are big? Why your ears are big? Have you ever thought of it? Why don't you have ears on the top of the head? Why your ears are curved? Have you ever thought of it? You think it is just by mistake? No, God doesn't do mistakes. So it's not just by mistake that your ears are curved. The reason why your ears are curved is because you cannot tilt your ears like some of the animals. And we find out that those animals which can tilt the ears are they are not that much curved. So the curving in on your ear is as a result of trapping this sound from any direction without tilting your ear. If someone calls from the back, you'll be able to know that this sound is coming from the back. If there's someone is calling you from the side, you'll be able to hear, you'll be able to know without even looking at that person. Why? Because your ear is trapping that sound from any direction. So we have this part, which is the outer part of the ear. We call it the pinna. So the ear is divided into 3 sections. It's divided into this part. So all this is outer ear. Then you have this, which we call the middle ear. And then from there to this, we call it the inner ear. So now let's look at it in detail. The outer ear. The outer ear has what we call the tympanum. The pinna. The pinna is this part which you see. So we call it pinna. And then it has this tube, which we call the auditory canal. Don't call it eardrum, call it auditory canal. Then we have this part, which we call, it's like a drum. Some books, they say eardrum. Don't call it eardrum, call it tympanum or tympanic membrane. Then we have the 3 bones, the smallest bones in a human body. The first one We call it Marius, Incas, and Stepes. I abbreviate it as MIS. Don't call it hammer. Sometimes they call it like that. Hammer, anvil, and then stirrup. Yeah? Yes. So you better call them Marias, Incas, and Stepes. That is scientifically. That is a KO, meaning that they won't argue about your answers. Okay, we continue. So these bonds, all these things they have, not these bonds, not these bonds, these bonds, these bonds, those are the three bonds. All these parts, they have a function. So the same way you see me moving, the same way the sound is moving. We have this window. Check the shape. It is oval. So it's oval window. Inside there, you have a fluid. That fluid is called the lymph. It has the endolymph and the perilymph. We don't need to know it now. But that fluid is the one which helps in the transmission of this vibration. And then inside there, we have this coiled part, we call it a cochlea. We will see what is inside the cochlea. And then now we have the nerves which come from this cochlea, and then they go to the part of the brain. Remember, this is a sensation. This is a sensation. So hearing is a sensation. Therefore, we will call it, it is being called, it goes to the cerebrum because cerebrum receives and interprets all the sensation. But there is this part we have not talked about, which we call the semicircular canal. So this is where balance is being done, meaning that your balance is as a result of your ear. So if you slap someone on the ear, then it means that that person will lose the balance. So don't do that. So don't slap people. However much it's so nice to slap someone on the ear, but you might lead to permanent effect of the balance. The person might lose balance permanently because we know you are enjoying slapping someone. Yes. All right. So meaning that the nerves which come from here, they don't go to cerebrum because the balance is not a sensation. Remember, balance and the muscle tone, equilibrium. We said which part of the brain is being controlled by cerebrum. Yes, is being controlled by cerebrum. So that it means that the nerves which are coming from the ear, they go to cerebrum. Now, we need now to understand the function of each. The function of each. All right. There is some few parts which we have not talked about. That is the eustachian tube. When you yawn, you feel that there is an air which is coming from the mouth to the ears. Yes, that air is there. It's going through the eustachian tube. Then you have the round window. It's round. That's why it's called round window. This one equalizes pressure from the inner ear. So now we need to know the function of each. Step 1. Pinna, the outer part. It collects sound and directs it. Corrects and directs it. So we want to see that word, it directs the sound wave. You see, it collects it and then directs it into this auditory canal so that you are able to go to the tympanic membrane. Then the auditory canal will transmit this sound wave so that it's able to hit the tympanic membrane. The tympanic membrane will vibrate. It's like you beating the drum. Yes, it will vibrate. And then when it vibrates, the vibration will be transmitted by these three bonds. That is the Marius, Incas, and the Stepes. I say don't call it hammer, anvil, and stirrup. Call it Marius, Incas, and Stepes. If you call it Hammer, Invel, and Styra, they might give you, but after a discussion. So it's better to use this so that your answer has not been discussed about. So now these bonds, we say that we call them oscos. The 3 bonds found in the ear, we call them what? Oscos. These are the smallest bonds in the human body. Yeah, that is the Marius, the Incas, and the Stepes. So these 3 bonds, you see these lines I put. It shows that is the outer ear. Then these lines, you see them here. Those are the middle ear. And then this line going like this, that is the inner ear. So this oscos also has another function. From transmission of this vibration into the oval window, it has another function of amplifying the vibration. That's why I say that it amplifies the vibration. It amplifies the vibration. Then now we have the oval window. It's like oval. What I showed you here, that's like oval. So it's oval window. The function of the oval window is transmit this sound wave into the inner ear. You see this blue color. I don't know whether it's blue or what. There is a fluid inside there. So the oval window will vibrate in and out, and then causing this fluid also to move. You understand? So now we have entered the inner ear. So now this cochlea, yes, it has what called the organ of Corti. You need to know that. The organ of Corti. Those are the receptors. In the eye, we call them photoreceptors. Those are the rods and cones. Those are the sensitive cells. Here, we call them organ of Corti. How does this thing look like? Basically, when you uncoil this, this is how it looks like. It will come like this. Ne? Inside there, it has sensory hair. Sensory cells, those hair, they have these cells. So now when this fluid is moving, it's going to cause this hair to move in and out. They move like this. Because this fluid is shaking. Yes. So when it shakes, then it causes, because they're sensitive, these cells, the impulse will be fired, and then it's being sent to cerebrum, because this is sensation, hearing. And then the excess pressure will go back. You understand? And that excess pressure will be trapped by the round window. That's why you're saying that it raises the pressure from the inner ear. When you absorb the excess pressure, it means that now you are releasing this pressure, which is from that side. So you can say it releases the pressure from the inner ear or absorbs the excess pressure from the inner ear. Yes. So now it means that this impulse, these nerves, yes, they will take the impulse to the brain. Which part of the brain? Cerebrum. We have seen it. So that's why. And then now, this pressure inside, it must be maintained. When it is maintained, there is this tube which called the Eustachian tube or Eustachian tube. I don't know how you call it. Eustachian tube. This is the tube which helps to equalize the pressure on the either side of the tympanic membrane. When you yawn, how do you feel? You feel that the air is in between. So it's like those people have ever gone to trains, the underground trains. Yes. Where there is a time where you reach, you feel like there's something which is hurting your ears. It's because of the pressure. What do? Open your mouth, talk a bit. When you talk, make a yawn. When you make a yawn, the air will come in, and then now it will be equalized. Meaning that now here the air is being sucked from this side. So it means that now once you open the mouth, the air will go through the Eustachian tube, and then it will make the air between the two sides of the membrane to be equal. And then that painful thing you hear in the ear or you feel in the ear yes, it will go. So that's the function of the eustachian tube. Equalize pressure on the either side of the tympanic membrane. We say that the cochlea contains the organ of Corti, which is responsible for converting the stimulus into an impulse. And then it moves these nerves, which we call the auditory nerves. Auditory nerves. These nerves. Yes, the one which is coming from the balancing part, semicircular canal, and the one which is coming from the cochlea, both of them are called the auditory canal nerves. However, the one which is coming from here, it goes to the cerebellum, while this one, it goes to cerebrum. So this part which you see, the semicircular canal, is the one which is responsible for balance. So you guys, you have to know this here. Take it slow, get it, make sure that you understand it. Step one, you need to know how to label. Once you know how to label, you need to know the function. If you know the function, then you'll be able to know the hearing process. So when we come back here, which we call the hearing process, is simple. Why? Because you need to know each and every part which is here. Yes. This is just trying to show you the internal part of the organ of Corti, of the cochlea, that the cells which I've tried to explain. Let's look at hearing process. You're going to go together. As I'm talking, you also talk inside your head. Step one, the pinna traps. We say that what's the function of the pinna? It traps sound wave and directs it into the auditory canal. Auditory canal will transmit the wave into the tympanic membrane. You see, I'm just talking about the part and the function, and then I'll bring, as I'm talking about the part and the function, putting them together, then it is forming the process of hearing. The pinna, the pinna traps sound wave, and it traps and directs it into the auditory canal. The auditory canal transmits this wave into the tympanic membrane. The tympanic membrane will vibrate. Once the tympanic membrane vibrates, it causes the oscos to also vibrate, transmitting the vibration into oval window. The oscos also amplifies the vibration. So now, the oval window will vibrate, causing the fluid in the inner ear to vibrate. You see? The vibration of the fluid in the inner ear, yes, will cause the organ of Corti, yes, to be stimulated, firing the impulse to cerebrum for interpretation via the auditory nerves. You see? That is the hearing process. Let me repeat. The pinna collects sound and directs it to auditory canal. The auditory canal transmits the wave into the tympanic membrane. So tympanic membrane is done, pinna is done. The tympanic membrane will cause the oscos to vibrate, you see? So it will start to shake. The oscos will will amplify the vibration and also transmits the vibration to oval window. I see. So oval window is also done. So now the oval window will transmit the vibration into the inner ear. You see, the vibration of the inner ear will stimulate the organ of Corti to change this stimulus into an impulse. You understand? And then which is sent to cerebrum for interpretation via the auditory nerves. That's it. So we have some parts which we have not talked about. We have some parts that is the round window and eustachian tube. So the round window, we can just talk about them just separately. Round window will absorb the excess pressure from the inner ear or release the excess pressure from the inner ear, while eustachian tube will equalize pressure on the either side of the tympanic membrane. We have not talked about the semicircular canals because semicircular canals is not hearing. That's the balance. We're going to look at it separately. So when you come back here on this hearing process, pinna traps and directs, you see, the sound wave into auditory canal. You see, tympanic membrane vibrates, transmits the vibration to oscos. That's the same thing I've talked about into the middle. Yeah, the oscos amplifies the vibration and carries this vibration in via the middle ear to the membrane of over window. You see, goes to the over window. So what's happening with the over window? Over window vibrates, causing the pressure wave in the inner ear. So it causes that wave, pressure wave, so that the fluid inside is able to shake. Then now the cochlea vibrates. Yes, remember those cochlea vibration, not cochlea doesn't vibrate. The vibration in the cochlea, yes, will cause the sensory cells that is in the organ of Corti, yes, in the organ of Corti to be stimulated. You see? And then now, the cochlea and the nerve impulse the nerve impulse is generated, meaning that the stimulus in the cochlea is converted into a nerve impulse. Then the auditory nerve will transmit this nerve impulse to cerebrum for interpretation. That's the hearing. 6 marks, you must know them before you approach your exam. June exams, people who are doing June exam as finals, people who are doing June exam as grade 12, not final exams as June exam. Please don't make a mistake to go there without knowing this. Or otherwise, you are losing marks. Losing marks. So that's why you see they didn't talk about the oval window, sorry, the round window and the eustachian tube. We said that those are just parts which are responsible for maintaining this process happening by round window absorbing the excess pressure from the inner ear, and then the eustachian tube to equalize pressure on the either side of the tympanic membrane. So guys, that is it. Hey, I'm seeing that the time is rushing us. All right. So let's do it together. Let's label, guys. Let's label. Okay, what is A?
Pinna.
Pinna ear, pinna. And then what is B?
Tympanic membrane.
Tympanic membrane. What is C?
Ossicles. MIS.
What is the middle part? Remember, this is Marius. This is Incus. And then this is Stapes. So they are looking at the middle one. So you can't say oscos. So if it was generalized as this, then you say oscos. But now they pointed on a specific bone, which is in the middle. So it's going to be Incus. Yes. What is D?
In what?
Semicircular canals.
D is semicircular canals. What is E?
E is cochlea.
Cochlea. Yes. What is F?
F is the auditory nerve.
Auditory nerves. What is G?
G is the
auditory nerve.
Eustachian tube.
Auditory tube. Eustachian tube.
Eustachian tube.
I don't know. Some people they call it eustachian. Eustachian tube. Yes, eustachian tube. Yes. And then what is H? H is this?
H is the
oval window.
The round window.
Check the shape. That is
oval window.
That's round.
Oval
So which
window.
Eh?
Round window.
Is it
round
oval?
window? Round window.
Round.
It's round.
No, it's oval.
Round or oval?
Guys,
Oval
this
window.
one is round. Oval is this. Check this one.
No, it's
Let me zoom it
oval.
out.
So it's
Let me
oval
zoom it
window.
out.
It's round window.
Don't you see
The
that
oval
this
window
is
is
round?
the
This is round. This is oval. So the answer is round window.
Okay.
But
Yes.
Round or
Yeah. If you check it again here, you are able to see that's what we said, that the upper one is oval window, and then the lower one is round window. It's round window because it's round. It's oval window because it's oval. Huh. Why do we call it a window? Because it is a gate from one point to. That's why they call it a window. Okay. Give the letter of the name. Okay, we're done that. Transmits impulse to the brain. Which part of the which transmits impulse to the brain?
F, the auditory
F,
nerve.
auditory nerve.
Which is the auditory nerve. Thank you, guys. And then allows pressure to equalize between the outer ear and the middle ear.
Eustachian tube.
Tube.
Eustachian tube, which is G. Remember, they said letter and the name, meaning that you have to get those 2 marks. Give only the letter of 2 structures in the diagram of the ear. When damaged, you would result in the following. Number 1, conductive hearing loss.
E coquês.
B. B.
B.
E.
E.
F.
B.
Yeah. Right, guys, when you say conductive, what does it mean? It means
It
that
allows
something
sound
which
to be transmitted into the ear.
basically the vibration. Where do we have vibrations? We find vibrations here. So any of that part, if it is damaged, so basically, since they put the letters, is B and C. Remember, they conduct. They attach to one another. It comes as a vibration. You understand? And then when they say now this, then it means that it is as a result of you can't sense the impulse cannot be transmitted. So it means that it's gonna be
F.
E. You understand? It's gonna be E. But still, because that's where it's being sensed from, but the impulse will not be transmitted. So also F. So they said give only the letter of 2 structures in the diagram of the ear. That's why you'll find out that it's B and C, and then here you'll see is E and F. E because it has the organ of Corti which senses, and F because now the impulse will not be transmitted in terms of an electrical message. Neuron, you see? And then this is sense. So the sensing is here in the cochlea, while the neuron is the F. What are they saying? And they're saying that middle ear infection are common causes. All right, we have not talked about the middle ear infections. Let's just talk about, okay. Let's talk about balance. Guys, are you ready for balance? I want to hear from you. Are you ready? Should we go?
Yeah.
Okay. Now here we need to listen carefully because we have new words which are involved. Among all the process of the nervous system, this one, they like it because they know that you fail it because you know that you don't remember the words. So this process is full of scientific words. Is it clear? It's simple, I did this Saidi mode of balance so that it's able to simplify your work. So at least you must know this Saidi's model of balance before you go to the what? To the paper. Let's start it. Balance. You see, balance is being controlled by the semicircular canals. In the semicircular canals, you see that these semicircular canals, they have a specific plane so that you are able to be detected in any angle you are talking about. So, balance. Balance is controlled by semicircular canals. We have two receptors. We have a first receptor, which we call the cristae, and then the second receptor is called maculae. We're going to ask you this. The first receptor is called cristae. The second receptor is called maculae. Cristae, maculae. Cristae, maculae. Cristae, maculae. Cristae, maculae. So you have to sing that so that it's able to stick in your head. You sing it, you write it down. You sing it, you write it down. You sing it, you write it down. You love it because if you hate it, it won't go into the head. And if you hate it, it's what they want to bring. And if they bring it, because you hate it, you're going to fail it. So cristae, maculae. Cristae, maculae. Those are the receptors. But where is the cristae? What is the cristae used for? Cristae is responsible for change in the speed and direction. When you change the speed and direction, change in the speed and direction, speed and direction, speed and direction, it is cristae. The position of the head, you can nod your head. How fast is it? It means that it is the cristae which is going to be stimulated. But if you change the position, if you, when you nod the head, you are changing the position, isn't it? Yes. But if you shake your head, you are changing the direction of the head. When you shake it, you're changing the direction. Yes. Well, when you nod it, you are changing the what? The position. So when you nod it, when you change the position, what is stimulated? It is the maculae. Okay. Now let's start again. You have the stimulus is speed and direction. The second one is position of the head. How many receptors? We have two. The first one is cristae, responsible for speed and direction, while the speed and the position is maculae. That is position of the head. Okay. Now you need to know where is this cristae found. It's found in the ampulla of the semicircular canal. There is a part called ampulla on the semicircular canal. So it's found on the ampulla of the semicircular canal. While the cristae, the maculae, is found in the circulus and the utriculus. Circulus and utriculus. Circulus and utriculus. Circulus and utriculus. I'm saying it so that it sticks into your head. You have to sing it. I repeat, you have to sing it. So, the ampulla of the semicircular canal, we find the cristae there, while the circulus and the utriculus, we find the maculae. Because this and this are receptors, they're going to change the stimuli into a nerve impulse. And then the nerve impulse will be sent to cerebellum. Why cerebellum? Because that is balance. How does it go there? It goes along the auditory nerve. Why does it go to the cerebellum? Interpret the information so that balance is maintained. Okay, now let's repeat. Let's now say, how do we write in the paper using the Saidi's model of balance. Step 1, you need to identify the stimuli. Step 2, you need to identify the crystal. Step 3, you need to know the location. Step 4, now you need to combine the two. You will say, change in the speed and direction of the head will stimulate the crystal found in the ampulla of the semicircular canal. Can I repeat? Change in the speed and direction of the head, yes, will stimulate the crystal found in the ampulla of the semicircular canal. That's step 1. You put a comma. Step 2, change in the position of the head will stimulate the maculae found in the circulars and utriculars. That's another comma. The stimuli is converted into a nerve impulse. The stimuli from here and here, that's why it's called stimuli, the plural of stimulus. The stimuli is converted into a nerve impulse, okay, which is moved along the auditory nerve. Where? To cerebellum. For what? For interpretation. So what? To restore balance. Simple. So your job is to know these words. Your job is to know these words. Changing the speed and direction of the head will stimulate the crystal found in the ampulla of the semicircular canal. Change in the position of the head will stimulate the maculae found in the circulars and utriculars. The stimuli is converted into a nerve impulse, which is moved along the auditory nerve to the cerebellum for interpretation to restore balance. Go for Saidi mode of balance. Yes. So guys, make sure, yes, if you don't have this booklet, take a screenshot. I've cleared it for you. Take a screenshot. Make sure. If there is any other word I should use it to mean that make sure you know this before you go to the paper. If there is any other word, emphasize that, use it. That's balance. When you come back here, the same story. The crystal in the semicircular canal, okay? Yes, you see crystal in the semicircular canal are stimulated by change in the position, by changing the direction of and the speed of the movement of the head. The maculae and the maculae in the circulars and the utriculars are stimulated, yes, by change in the position of the head. The crystal and the maculae, yes, convert the stimuli into nerve impulses, which is transmitted or which are transmitted along the auditory nerve to cerebellum. Interpretation, yes. For interpretation, the cerebellum then sends impulse to muscles so that balance is restored. You see that what is here in different color is exactly what is being written here. So you can use your words as long as you don't miss any of these words. If you miss any of these words, then it means that you are missing a mark. Yes. I know it might look complicated. Doesn't look complicated if you keep on repeating it. Make sure that you repeat it. You have enough time to do this. Yes. I was supposed to rush this, but I've not rushed it. So because sometimes rushing does not make a benefit to a student if when you just want to rush, finish up a specific part you wanted to finish. I hope you guys, you have got it. Let's talk about, let me just emphasize this before I talk about the effects. When they bring a question, how do they bring a question? They can bring an open question whereby they ask you to explain balance, or they bring a scenario that Zintle was playing netball, tried to shoot the goal, and then suddenly lost balance and then fell. As he's falling, the ball is coming back, falling back, wants to save the ball, and then also try to grab the ball. You see that you are changing the position and you're also including the speed. Explain, how did Zintle try to restore the balance after reaching the ball? So it means that you're going to have to explain the speed and direction, and then you talk about the position of the head. At the end, you have to link this to catching the ball. Do you understand? For interpretation, so that the ball is catched and then balance is restored. So you have to explain depending on the scenario they have given you. Sometimes they give you a scenario whereby you only talk about the speed and direction, meaning that you explain that. If they only ask you a question which involves the speed and direction, meaning that you only explain this. And if they ask you something about position, then it means that you explain this. In other words, this is combined. See, you have to talk about whether it's speed and direction or position of what? Of the head. While this, because only for speed and direction, should only be talked about when you're talking about speed and direction. And then this, only for position. But Mina, I will advise that if they bring such question, because they don't bring this question and they say list 2 or list 4, just explain everything, they will find out the correct answer from there. Obviously, they cannot lose it. They cannot fail to give you the mark because you talked about this. Unless they are specific, they say give 2 points. But if they say explain or describe, describe everything, the answer. If you don't know, if you are confused, you see that things are not right for you, just do everything, because this is a description. When we are marking, we are looking at where is the correct answer. Even if you have written 10 pages and the answer is there in the middle, we look for it. Yes. So I think I'm clear there. Let's just talk about explain the ways in which A is in diagram structures. Yes. So you see, now you can also practice that question. I'll paste it in the group so that you're able to do it. Hearing defects, middle ear infection, meaning that here you have problems with the did you know that many people are deaf? If you have said, yeah, you're also deaf. Yes. Because deafness does not mean you are 100% deaf. Hearing losses. If you have hearing losses, then it means that you are deaf. What is happening? Why are you writing? Why are you writing on my screen? Stop it. We can call it. What's happening now? Is it my system or someone is? Kutlwano, are you the one writing?
No.
Okay. Okay, I'm seeing something being written on the screen. All right, it's fine. So deafness, we say that deafness can be, you can be deaf, partial deaf, or completely deaf. Okay, so middle ear infection, first of the things which causes this deafness is, number 1 is middle ear infection. This is excess fluid in the middle ear. What causes that is a pathogen. A pathogen can be a bacteria. Remember, a pathogen is a disease-causing organism. Anything which causes diseases is called a pathogen. So in this infection, in most cases, is caused by pathogen. And guess what? This pathogen is so bad in such a way that it's gonna cause the ear start accumulating a fluid, a pus. As that fluid is smelling badly. So it means that this part, this here, the fluid accumulates there. You see? Now it's gonna start to come out of the ear like this and is smelling. I think you have ever seen such people. It is not because of the koloshi. No, is because of the bacteria which is causing this thing, the inside part of it to rot. Meaning that the inside part is rotting and then the pus is coming out. Is it treatable? Yes, we can cure it. How? You insert a structure called a grommet. By inserting a grommet, it looks like this. The grommet looks like this. It has a hole inside and it has two parts. So what we do is we put it in here. We cut your tympanic membrane. This is a tympanic membrane. We cut it and then we insert this grommet so that it connects this side and this side. And then now the pus is able to move out. Number two, because this is caused by a bacteria, so we apply what you call the antibiotics. So antibiotics will work there. So we insert the grommets. Number two, we apply the antibiotics. This can cause hearing loss. Number two, deafness. It could be a partial deaf or it could be a total deaf, partial deaf. What causes this? Some of this is caused by not cleaning our ears. Like too much wax in the ear. You have ever seen people having a lot of wax in the ear? It can be caused by that.
Sir?
Number two, it can be caused by injuries of the part of the ear or the nerves or part of the brain responsible for hearing. In this case, that is cerebral. You have
Mr.
got an
Said.
accident and this accident has caused you to lose these, or these parts are damaged.
Sir!
Yes.
But why is it told that we shouldn't use earbuds to clean the ear because it pushes the wax further? But you're saying if we don't clean it, it gets damaged.
Well, it's not basically it's gonna get damaged, it's gonna get blocked. So these you call them those earbuds is because if let me show you here. Let me use a bigger structure of the ear so that you're able to see what I'm saying. If this is your ear, now you put your earbud here, and then now remember the wax is here, ne? In science, we call it cerumen. I don't know why these things are happening on my screen. We call it cerumen. So now when you put the earbud, you are pushing this cerumen to the tympanic membrane. Meaning that now it's going to accumulate here. You see? It's going to accumulate there. So now when you push, yes, some of it will going to be pulled out, but some of it is going to accumulate here. And once it accumulates on the tympanic membrane, the tympanic membrane will not vibrate. That's why it's not that good. So you can clean them. Eh? I only know from my side as a Muslim that we're supposed to wash our ears 5 times a day. Every single day, at least a minimum of 5 times a day. So, meaning that your ears are supposed to be clean. But still the wax is still there. Small. Why? Because this wax helps us to trap some of the insects and stuff. You understand? So if you don't clean your ear, this wax is going to accumulate and then it blocks. Once it blocks, it means that the sound will not go in. properly. So you're going to have to suffer from what we call partial deafness. Sometimes this works in science, we call it cerumen. 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 I don't know whether you can see it. Yes. It's cerumen. Sometimes we call it like that. When you see it, just know that that is the earwax. How do we sort that out? By removing the earwax. Some people, they even go to the doctors to remove those big. Go to YouTube and then you watch, you'll see those big earwax. There's some also in Thundereduc. I think there are some videos we have already uploaded which show those earwax which are being removed. So another thing is hardening of the ear tissue. It could be like the oscars, it can no longer vibrate. We saw it that it can also lead to conductive hearing loss. There is no conduction of this vibration, meaning so that it means that now the hearing loss is gonna happen. So how do we solve that? We apply the hearing aids, something which is gonna help you to hear. You have ever seen people having this? They bath with it, they sleep with it. Yes, it's just to help them to amplify the vibration. Number two, if it is your cochlea which has been damaged, then you have to do the cochlear implant, like this kid. They drill here in your head so that they reach the cochlea. So this one is just a tattoo. The dad is showing them the kid that no, you are not alone. Yeah, I'm also having that. You understand? Just not to bring that. They call it what? They call it stigma. Yeah, in them they say that no, they become depressed. That okay, it's only me. What? Yeah. So he was doing that. It's just a tattoo. But you have ever seen people having? They drill it. It's an operation. So meaning that now these parts are no longer working. They're going to have to use this, and then the impulse will be sent to the ear, to the cochlea direct. Without these oscars and stuff, all those things will no longer be used. So guys, basically that's the ear. You need to know the structure of the ear. You need to know the function of the parts. You need to know the hearing process. You need to know the balance. You need to know the defects of the ear.
Sir,
And once you know that, then you are able to know.
sir,
Yes.
can I please ask what is a cochlea?
What's the cochlea?
Yes.
We have been talking about the cochlea.
Yes. Can you please repeat?
I'm not going to repeat everything, but cochlea is this part. This is the part. This is the cochlea. So that's where the organ of Corti, that's what we say that the organ of Corti, the sensory cells of hearing, that's where they are located. It looks like this. Yes. When I come, yeah, I think when I come, yes, when I come to schools, yes, I think I'll bring my this ear, and then I show you exactly what is happening, how is the transmission being done, and those people are attending from Thundereduc Direct. I'll also show you. Yes, I will show you from here, and then those who are attending online, there is, I don't know, maybe I will just make a small video and then I'll show you how using, but you might not see okay, I'll have to show you, but if I don't, still you can also check it from some videos on YouTube. You can also find them, but maybe I'll try to do it so that you're able to see it physically. I would have showed it to you, but I have to go to the lab and then pick it. And the guy who is in charge of that is not around. So, guys, I think that is it. That is the ear. The next topic is supposed to be human endocrine system. I'm going to give you homework on this. what is endocrine system, hormones. So we need to know this. There is nothing new I'm going to talk about here. I'll just explain. But you have ever heard about hypothalamus? You have ever heard about pituitary gland? You have ever heard about adrenal gland? Have you ever heard about pancreas? Have you ever heard about ovary? Have you ever heard about the testis? You have ever heard about the estrogen and progesterone? Have you ever heard about progesterone? So it means that now I'm just telling you to go back and read about it so that when I come back to teach it, you are able to remember. You have ever heard about the glucagon and insulin, that is grade 11 work? If not, these are the people who are suffering from sugar diabetes. It's because of this. Adrenaline, when we talked about it, when you are getting scared, so adrenaline is going to be what? It's going to be much so that it's able to prepare you for emergency. The only hormone which I say that maybe you have never heard about is aldosterone. Those people who did grade 11, I think you did it. You did. And then this one, thyroxine, you've never heard about it maybe. ADH, we will also see it is responsible for water, amount of water in the body. So the pituitary gland, when we are talking about the brain, we said we're going to look at it here much more. Even the hypothalamus, we're going to look at them here much more because of the different hormones it produces. But among the hormones, we talked about follicle-stimulating hormone from reproduction. We talked about luteinizing hormone still from reproduction. So you can read about, so basically you can read about the menstrual cycle because the menstrual cycle involves FSH, LH, estrogen, and progesterone. So you can read about them before we come back for the next class. Then you are able to, at least you can link. Here is just to talk about the pattern function. And what if this does not, you don't have it in the body, what is the consequences? That's it. And then after that, how do they work in the negative feedback mechanism? I'm just doing what we're going to cover in the next class. So how do you see that if you don't have this, you're going to suffer from goiter, something like that. If you have too much of it, it's hyperthyroidism. If you have less, it's hypothyroidism. So that's it. And then homeostasis, what we're going to cover in the homeostasis is that now it's the same glucose, the same hormones we have talked about, is the insulin. What will happen if it is too much? How do we bring it back to normal? If it is too low, how do we bring it back to high so that it goes back to normal? So those are the negative feedback mechanism which we talk about. We're going to talk about carbon dioxide, and then now we're going to talk about the salts. The salts, if too much in the body, people who eat too much salts, what is the effect? Yes. I talked about, and then the skin. We'll talk about the last organ as the skin, and then that is keeping the internal environment, keeping the temperature constant. And once we're done with that, then it means that we are done with this topic. This topic is not that big, it's just an hour. It can be covered because I'll just show you the techniques we use. If you are looking for salt, you can do for water, you can do for thyroxine, you can do for glucose, all of them. It's just the same technique. So see you in the next class. Guys, I'm going to stop here for today. I thought I'm going to finish all of them, but the time has